All the work I'm going to show today is actually unpublished. It's work that myself and my young lab have been working on since we got our Bruker 2P+. And we're pretty excited that the data we're getting we hope to turn into several publications. So hopefully the story comes across today. Okay, so many of you know this kind of image, very familiar type of data, but this is just your standard two-photon resonant galvo scanning calcium imaging. This happens to be ferret visual cortex, which is the model system that we use here in the lab. But I like this image because it really shows the dynamics of a circuit and the cellular populations of cells. We spent a lot of time as neuroscientists really trying to understand the properties of cellular circuits, trying to understand the statistics and understand what information you can encode or decode from them. But I've been really interested in understanding what makes circuits work. And of course, the underpinning to all the circuits and each one of these cells, so everything you see here, you're mostly seeing these cell bodies, but underneath, within these cell bodies are also many other processes that these neurons have, mainly dendrites and inputs that arrive onto those dendrites. So each one of these cells actually receives a huge collection of inputs, many from their surrounding neighbors, but some from other brain areas. And the collection of these inputs give the cells their operation. So each cell actually has kind of a synaptic input population or a network that it interacts with. And so really a single circuit or a network of cells is really a multiplexion of many, many different networks that are working together for brain function. So, historically, I've studied this using synaptic calcium imaging, which is really just conventional two-photon imaging with a little bit of a viral trick, where we sparsify the indicator of interest, in this case, 𝗔𝗕𝗦𝗧𝗥𝗔𝗖𝗍, and thus we only have very, very sparse expression of individual cells, and so we can resolve things down close to the point spread function, and that being little processes on the dendrites, such as spines. So like I mentioned, the animal model that we use in the lab is the ferret. This is a very great model of the early visual system. It has components of the early visual system that's more similar to humans and primates than they are mice. In particular, and this is going to be important for today's talk, within the visual cortex, you have this functional topography. You have this columnar organization of features that are encoded by the neurons, so that such that neurons nearby each other share the same feature selectivity. For like 10 years, I basically spent a lot of time in my postdoc lab, David Fitzpatrick, trying to understand the excitatory networks within cells. So I did a lot of this spine imaging, and this is just an example of what spine imaging looks like. I'm sure maybe many of you have seen this kind of data before, but basically by presenting different visual stimuli to the animal or recording from a small compartment on an individual cell, we can see small isolated events, both in individual spines, that's the bulb-like structures on this branch, as well as global events that are due to backpropagating exponentials and other dendritic nonlinearities, such as regenerative events. So you get a lot of information, and you can analyze this data, and you can look at the individual responses of inputs and compare them to the outputs. So the output would be the calcium responses from the cell, the top here, the inputs being all of the spines that you can see down the branch. And we did a lot of studies. This is just one example of many, many studies where we found that there's this huge functional diversity of inputs. And for many years, we have, and more people have actually found this. And for many years, we basically are trying to understand what it means to have functional diversity within a cell and what this means for the cell's computations. But there's a much larger question that you can ask, which is like, what does the excitatory network do? I mean, we know that we have all these inputs on cells, and they range anywhere between 5,000 and 10,000 excitatory inputs on a given large pyramidal cell in the cortex. So what is the purpose of all of these inputs, and particularly within this large functional organization? And so this question and a few others led me to move my lab in a different direction to design new experiments to address a new set of questions. So in particular, what I wanted to know is, what do these recurrent excitatory inputs do? There's many of them. They seem to have a particular function. What is it? Also, by measuring the inputs of cells on cells, you don't really know how neurons interact because you're just seeing this like postsynaptic effect on the cell. So another thing that we'd like to know is how do neurons act during processing, during sensory processing. And then a really big missing piece of this whole puzzle is that there are also inhibitory neurons. And so although synaptic imaging has been really valuable in learning about excitatory networks in the cell, we also know that there are surrounding interneurons. They make up about 25% of the cells in the cortical circuits, but there is no morphological or biochemical component that can give us a sense of what the inhibitory inputs to cells are, nor is there really good tools for mapping, or at least there hasn't been very good tools for mapping inhibitory networks and understanding the contribution of interneurons onto excitatory neurons. So the question was, okay, how can we address these different questions? What's the technology that we could use? So that's what I turned to all optical calcium interrogation. So this is using two-photon calcium imaging in conjunction with two-photon optogenetics, either with a scanning or scanless system. So in this case, we're using the Neurolight system to both stimulate single neurons and multiple neurons. So the way this works is that we have two imaging systems. One that is doing the stimulation, one is doing the actual imaging. The imaging path is around 940 or whatever your particular indicator of interest is, in this case GCAM. And then in our hands, we're using a somatically localized two-photon opsin called Chrome. There's many variations of this now. We've got Chrome 2.0, Carmine from a variety of different labs, but they all serve the same function, which is that they're a big cation channel, and when you hit it in the right way with to absorb long wavelength light, you can get these nice, beautiful responses as shown here on the right. Okay, so there's actually a bit of history behind the idea of doing influence mapping to look at the interactions between cells and map how they're related to each other. And one of the earliest studies that I know of is from Alex Kwan and Yang Dan, where they actually kind of pioneered this approach a little bit, not doing all optical, but doing some kind of a mixture of the two. So here they were patched or cell attached onto a given pyramidal cell shown in the blue, and then stimulated it with electrical current in order for it to drive spikes. And then they were using old school imaging, so this is like OGB, and looked at changes in calcium that would be due to stimulating a given pyramidal cell, and thus they could map, look at the interactions between that cell on all the others. So this is the strategy that people have used, and now we've taken it further to doing all optical. Now, there is several studies that have done this in mouse V1 with some differences that I'll explain in a minute, that have revealed some potential resulting interactions in the excitatory network. So previous data from Chris Harvey and then subsequent studies from Hillel Adesnik and others suggest that in mouse V1, at least, when you stimulate an excitatory neuron and look at the activity of surrounding excitatory neurons, there is this Mexican hat-like connectivity and competitive interaction. So basically what they do is they stimulate a given cell, they look at how the activity changes, and then they look at that change of activity and they use that to say that calling the influence, and they look and see whether that is excitatory or suppressive. And so the Mexican hat component here comes from looking at this curve. This is, if you just pay attention to the orange line, so basically nearby, so within distances of 0 to 100 microns, there's positive influences, and then it drops really far down, goes suppressive, and then kind of comes out back up to about 0-ish, around 250 microns. And so this is kind of the Mexican hat-like structure that people would see. They also did see that if you look at the correlations between cells, their cells are highly correlated with each other, do also have positive influences. So this would be sort of like functional coupling between neurons. So there's a little bit of evidence for competitive interactions, the suppression, but also some facilitation. And it's unclear whether this applies to other animal models and how this would work in a visual system that has topography, a visual cortex that has topography. There's another aspect of the study that I was a little bit, that I thought could be built on a little bit, which is the measurement of influence. So in these studies, people kind of measured influence by more or less using a z-scoring method. So you take like the mean change in activities and divide it by the standard deviation. And that was something that I wanted to build upon in our study, and it seemed to be really critical for how we analyze our data. Okay, so as I mentioned, there's a big difference between mouse and ferret. So in mouse V1, the cells, they have this kind of salt and pepper-like organization, which is just to say that cells nearby each other don't necessarily have the same property. They don't care about the same features in visual space. Whereas in ferret, you have these columns, and so cells that are nearby may have the same direction preference, same orientation preference. They generally have the same receptive field structure. So if you take the ideas from the mouse, it's not really clear how they would layer into the ferret, where you have this topography, this local topography. And so that was one of the reasons that we really wanted to do, basically first just replicate the same experiments that Chris Harvey and Hillel Desnik's labs have done. Okay, so here is the experiment in question. So the way the experiment works is that we express the indicator and the opsin in an animal. We place a window, and then we identify target neurons for photostimulation. So those are the cells in pink. We also identify a bunch of sham locations, and so typically we choose regions that are out of focus or blood vessels, and these are going to be areas that we're going to stimulate that we expect not to have any effect on the network, but we can use to calibrate our measurements. And then we're going to present a bunch of different visual stimuli to the animal. These visual stimuli range in terms of the direction that they move, as well as the contrast. So higher contrast means that the stimulus is stronger, and a lower contrast means it's weaker. So that's kind of shown here in B. And we do many, many different combinations such that we present, do about on the order of around 5,000 trials or 5,000 to 10,000 trials per experiment, with the idea that we want many, many combinations of every possible, many trials of any possible combination between stimuli and target, so we can look at the total influence across the different stimulus states. So here's just an example cell that we're stimulating. We get very nice, reliable photostimulation over many, many trials for individual cells, and there's a non-target. The other thing that we do in our experiments is that we try to titrate the photostimulation power. So typically we identified a stimulation protocol that works the best with about 250 millisecond stimulus duration, multiple pulses on the cell, to get reliable activation, but we don't want to create phototoxicity, and we want to limit the amount of laser power that we're throwing on the sample. And so we do kind of measure these power response curves and look for a regime in which we are activating the cell, but not at saturating the cell, because once we're saturating, we're basically just kind of hammering the cell without and putting more extra power into the system needlessly, because we're not going to get any more bang for our buck. So we try to find this ideal stimulation suite, and this is for a single cell. This was with the spiral scanning system. It's about between 20 and 40 milliwatts average power. We've actually got these numbers down quite a bit now with dialing in the SLM and using a couple of new indicators. Here's just an example cell. So this is on the left is a projection of the average activity within this field of view. You can see we've got very nice, dense expression across the entire field of view. And the single target cell in the center that we've chosen, you see that it's nice and red, so we've got this strong activation of this cell. So again, these are all excitatory neurons that we're targeting here, both with GCamp and for photostimulation. Okay, so there was one more piece that we needed to build that we found to be very critical, and that was how we're going to measure influence. And I was a little bit unsatisfied with the conventional methods that people were using, so we turned to using a generalized linear model to basically explain away the variance of each individual trial-by-trial responses by fitting these filters and trying to dial in the model to both basically fit filters to the tuning, that is the stimuli that we're showing the animal, as well as other features like neural pill fluctuations and global signals, and as well as the optogenetic input. So we have a whole bank of filters that include the sham locations, the optogenetic targets. We sum all those together, we pass them through an exponential nonlinearity, add some Poisson noise, and then we dial in these features to predict, try to get our best prediction possible of the real data. And so once we have a model that works pretty well, we can measure influence by actually just removing the optical target weight. So what we do is we generate our prediction, that's shown here in orange, and sorry for the low resolution image, and then we remove the optical, the weight, we set the weight for a given photostimulation target to 0, and then we take the difference between those two, and the difference in predicted spike rate is now our measure of influence. And so what I like about this method is that we're regressing off all of the correlated stimulus variables. We're regressing out the co-fluctuations with network and neuromodulatory states and stuff. And we're trying to isolate purely just the effect of stimulating a single cell within the network. Okay, and this works really well. We get many cells that have influences measurements. We also can use this model to get a statistical significance on the influence. So what you're seeing here is the influence targets and the influence target map for a single target cell shown in the green. Blue are cells that had suppression. Red are those that have activation. You can see there it's quite messy, it's quite diverse. And then there are many cells that are white, and those are ones that didn't have a statistically significant influence. So that's why we developed this method, and now we're going to use this method to look and see how excitatory neurons actually interact with each other. Okay, so the first thing I'm showing you is a measurement of influence, excitatory influence between other excitatory neurons as a function of cortical distance. So on the left is a distribution of all influences that we have in our population. You see it's kind of centered around 0, but actually you get large negative ones, you get large positive ones. The total distribution is actually biased a little bit towards suppression. So the little triangle there is shows that the median value is slightly negative. Note that the axis is log. And then what we did is we took pairs of cells, so target, non-target pairs, and then we calculated their distance from each other and measured the influence for those that had significant influences. We also were very careful to exclude making measurements around a given target within about 20 microns because we don't want to basically have any photostimulation artifacts in our measurements. So what we found was that by looking at these pairs of neurons is that pretty much all interactions are suppressive. The suppression falls off with cortical distance, so the ones that are most nearby show the least amount of suppression, but overall we have wide sweeping suppression across cortical distance that continues to get stronger and stronger as you move farther away. Now, what was really interesting is that because we presented all these different stimuli and with our GLM measurement, we actually could look at influences on a stimulus by stimulus context. And so we could separate and compare low contrast and high contrast stimuli, so those that are stronger and weaker. And we found something very different for the low contrast, in that the suppression was much weaker, particularly for the tail end, where you actually started to see some facilitation. So the differences in the influences and the interactions between excitatory neurons strongly modulated by contrast. Okay. So like I said, there's this topography of functional similarity in Phe1. These cells are share the same feature preference between their neighbors. One very good way that you can measure this is by looking at the noise correlations. So that's basically looking at the fluctuations, the noise fluctuations on a trial by trial basis and correlating them between two neurons. Those that have high noise correlations with each other tend to be cells that are more likely to be connected and such. But in ferret, you can actually see here on the right that the noise correlations that we measure between neurons fall off with an exponential decay that has the feature of the size of a column. So basically about 250 microns, the cells don't have the same, noise correlations kind of go into no different than random. But this is a reflection of the columnar organization, the topography, and also is a good measurement of correlation. So we wanted to know how this influence is compared between cells that are functionally similar, near and far. Okay, so the first thing that we did is we just said, let's look at cells that are very nearby our target and compare low and high contrast, and then look and see what happens as the cells become more correlated. So basically the ones that are correlated would be ones that are most likely to be part of a column or a functional column. And here we see a really big difference. So at low contrast, you actually see that there is strong facilitation or amplification between excitatory neurons that are nearby and correlate with each other, while you maintain the suppression at high contrast. So it seems that contrast is actually modulating the regime that the cortical circuit is operating at. And then as we move out farther and farther in distance, we see that this relationship shifts. So we still see that there's this difference between low and high contrast, but as you move and look at pairs of neurons that are farther and farther apart, you see that the more distal pairs are dominated by suppression at high contrast. So it seems like there's different roles for neurons at different distances in terms of how they shape each other's interactions. And so this result is very interesting, very surprising to us, but also suggests us something quite interesting in that contrast is actually modulating the state of cortical processing such that neurons that are more correlated with each other at low contrast are going to be in this amplification regime. So the cortex might be trying to amplify weak activity, and weak sensory stimulation, and when you turn up the sensory stimulation to be strong, you go into more of a suppressive regime. And this might be the competitive interactions to explain away or reduce redundancy of the neural code. And so if this was the case, you should see this on the level of information content within circuits. So the next thing that we did is we actually tried to get at that using a multi-class decoder. And so what we did is we took the activity of all the neurons across all the different trials, and we took only the sham trials, and then we trained and optimized a multi-class decoder. In this case, the best one that worked for us was a logistical regression multi-class decoder. And then we took each individual photostimulation targets trials, and we tested the decoder and said, okay, does the decoder get better or worse but after being trained on sham? So basically the decoders only train on data where there was no photostimulation of an actual neuron, and then we test on data where we actually photostimulate a neuron. So basically, if we look at the sham cross-validated error, we see that the error is reduced from low to high. This makes sense because there's the responses of neurons in low contrast regimes are noisier and they're less reliable. So that kind of gives us a little confidence that this thing works. But what was really wild is that when we looked at the high contrast stimuli, we saw this negative relationship. So basically, on the x-axis, I'm plotting the average target influence on the surrounding network. And on the y-axis, I'm plotting the change in performance of the model. So this is comparing the model error on the test compared to the sham optimized training. And essentially, the model got better for trials in which the photostimulated target actually drove suppression. So if the average influence was suppressive, there was more likely that the test configuration of the model was doing better. And then we compared we looked at the low contrast, we actually see the opposite effect, and that the targets that actually are having more positive influences, so potentially amplifying responses, are actually causing the model to perform better. So it seems like the decoder is giving us some evidence that there is this switching of regimes from suppression to amplification between specific neurons with the change in contrast. Okay, so you might be wondering, where is the source of this excitatory difference suppression? Well, obviously this suggests that excitatory neurons are primarily not maybe driving each other, but maybe the dominant effect is sort of suppression via driving inhibition. And although it's quite difficult for us to actually measure the E to I connection, we do have the ability to actually do similar measurements of inhibitory neurons. And so we've actually done a bunch of experiments now where we photostimulate inhibitory neurons to look at their effects on surrounding excitatory neurons. And so this is a similar experiment. There's a couple little tweaks here because interneurons are a little bit harder to drive. They have a little higher firing rates, but more or less kind of got this to work. And we use the same strategy. So now I'm plotting the influence as a function of distance for the E to E interactions. So the arrow here is supposed to just describe interactions. It's not supposed to describe connectivity. And I'm now plotting on a different axis so you can compare when we do the same kind of measurement for I to E. So presumably these would be interneurons that could actually drive excitatory suppression. We see large suppressive effects. So much stronger than the E to E interaction. So this seems like this might be one of the sources of how this is all working. We also see some very interesting interactions with noise correlations. We actually see that driving inhibitory neurons can cause basically positive interactions, positive influences between correlated pairs, and this presumably must happen through disinhibition. So it's for this kind of change at the high contrast, this would mean that inhibitory neurons must be synapsing onto other inhibitory neurons, disinhibiting them to allow for more a rebound from that inhibition and to see these positive interactions. And of course, we can actually measure that too. So we've been able to measure the eye-to-eye interactions as well, trying to build out the full recurrent set of interactions. So, okay. This is the inhibitory story is still ongoing, and we're working to try to understand it more deeply. It's definitely more complicated than the excitatory network story. The other thing that we're currently working on that I'm really happy that we finally got working very nicely is we're doing these same kind of experiments now with ensemble activity, and we're both not only comparing how the networks interact when we present clustered activations versus dispersed activations, but we're also doing this with different numbers and ramping up the numbers. So all the data that I've shown you today is all stimulating a single neuron, and that's obviously pretty unnatural. It's not really how the brain behaves in the real world. And so here we're kind of starting to present more naturalistic input patterns and matching what we see within the columns, as well as playing around with those different parameters to see how the network interacts. And already we're seeing very interesting types of behaviors. So when we activate a bunch of excitatory neurons within the same column, the primary effect of that, maybe not surprising now with the data that I've shown you, is that you see suppression. So here on the left is the pattern. There's the average activity of those cells in the field of view. On the right is the average responses of the targets and the non-targets. And basically the non-targets are mostly suppressed. You see this rebound, actually that's the rebound right after photostimulation is finished. When we turn we gate the SLM, and you see this rebound excitation that's due to the suppression. But then when we present this dispersed pattern, we see something very different. So there we see that we actually can activate off-target neurons, and we see a lot of excitatory interactions within the circuit. So these different patterns we're now playing with this. We are able to actually see test predictions from mouse V1 due to some recent papers from 𝗣𝗥𝗘𝗦𝗘𝗡𝗥 that actually show very similar results to the kinds of things that we're seeing. So it's going to be really fun to try to figure out the similarities and differences between our model and theirs. Okay, so I'm going to give a summary, and then I have like 2 more slides about some work that I'm very excited about that we're also working on in the lab that's kind of new stuff. So today I showed you that excitatory cortical neurons drive large-scale suppressive interactions through and within excitatory networks. I showed you that sensory stimulation strength shapes the current interactions. It seems to switch circuits from an amplification to a suppression regime. And also we found that inhibitory interneurons can supply large-scale strong suppression onto excitatory neurons, but also suggest that there's lots of disinhibition within these recurrent circuits. So we didn't quite understand what excitatory cells are doing. It seems like even though you have these large collection of excitatory inputs on cells, when you activate lots of excitatory inputs or even individual excitatory inputs, the main effect is suppression. So I think that a lot of the data that we have is suggesting that the connections to and interactions with excitatory neurons onto inhibitory neurons is a really big thing that we need to study and understand that might be more more important in understanding the larger role of cortical circuits in general and how they process information. Okay. So in the last few minutes, like 2 minutes, I just want to talk about this other project that I'm very excited about in the lab, which I think could actually benefit from some new technologies from Bruker. And that is development. So for many years, I've wanted to study development. The ferret is a very traditional model of studying development, and used extensively by some labs, including my postdoc lab. And it's been for a long time I've been wanting to know how I can get involved. And basically it came from realizing that the actual development of vision and visual behaviors have not really been put together in a good way before, and that actually development is a recurrent and dynamic process in itself. I think that if you go to the textbooks, you would see that the classic model of development is moving from is sort of this linear sort of thing where animals experience the world, it drives activity that then shapes neuronal structure and function that kind of just is made concrete, and then that is the way that the system is wired. But in reality, there's this feedback where structure and function dictate activity, which controls behavior and how you move through the world. So it's actually this cyclic loop that moves through development and through time. And so I really wanted to measure that very carefully. And of course, we had to start by understanding what ferrets do in general. And this was not something I'd ever done. So what we very first did back when I first started the lab is we just put animals in the box around eye opening and asked what kind of behaviors they exhibit. So basically 3 days after eye opening, in which systems are highly dynamic and highly engaging in tons of plasticity, the animals basically don't do anything. They're not very engaged. They're not moving their eyes very much. They're not chasing objects. But then just 9 days later, so 12 days after visual experience, the animals are pretty damn good at chasing cat toys, engaging in hunting-like behaviors. They have a lot of natural behaviors that are just wired up and ready to go really quickly. So within 2 weeks, you've got this very fast onset of this very complex visual behavior. So we now built a set of systems to track this in real time. We have a behavioral arena with doing 3D tracking of kinematics and the motion of the animals. We have a magnetic system where we can do simulated prey. We also have built a custom eye tracking system that is on a head-mounted system so that we can track where the eyes are looking and what how the animal's actually seeing the world, with the idea that we can actually measure precisely what the visual input to the eyes are, to the brain of the animal from the moment that they open the eyes. And what obviously we need to do next is now couple these measurements with measurements of neural activity. That could be either done with physiology, but I'm also interested in looking at miniature 2P and other technologies coupled with this to track longitudinally over time from the moment of eye opening while measuring all of the behavioral and components of the animal. Okay, so with that, I'd like to thank the people who made this work possible, my lab, as well as the many collaborators that I have both here at Anschutz and across the United States and international, as well as my funding. Cool, thank you very much.
All right, that was great, fantastic stuff. Thanks for sharing that. I appreciate seeing unpublished data at talks. You know, it's not always the case. But yeah, it's really intriguing stuff. I actually for the record, everyone, feel free to fill out any questions using the integrated questionnaire box in the GoToWebinar panel. Right now, though, I don't have a serious science question right now, but kind of a methods question here. I noted that early on you said that you're using Chrome, and I kind of was just curious to know if you wanted to highlight any specific reason why you use that over other options, or if is it wavelength? I get asked these questions a lot, and I'm always curious to hear from people who are actually doing the experiments.
Yeah, there's a wide variety of vectors out there now. There are subtle differences between them. I'm pretty familiar with the whole cast of characters. There's also Chrome 2.0 now, which is mutated to have a much larger pore. I have not personally tried that one yet. So the reason that we use Chrome is that when we first started doing these experiments, we kind of threw the kitchen sink at our prep to find an opsin that would give stable expression via virus, where the cells were healthy and you got reliable activation for hours over long or even over days. And that's not the case with all indicators. So there's always a trade-off, right? Wavelength is a little bit. Not too much. Most of them have a long tail of absorption. So, we're not changing the wavelength too much. If you're using a Monaco, it's set. We are playing a little bit of games where we do stretch the Z profile of the optogenetic stimulation as well, because we want to sort of spread that laser power over the neuron. And we just do that with adding some glass or something, but you can do it by shaping on the SLM path too. The main thing is that the larger the pore, the more sensitive the cell is to photoactivation. But that has two caveats. So first of all, the two-photon absorption of these tools are very wide, right? And so most people actually try to do their imaging stimulation around 900, 920 nanometers to avoid activating those sensors. So if your prep has very low expression of GCamp and you have to use more power, you actually start seeing you just start introducing excitatory activity throughout the whole thing, and you'll see kind of like epileptic events and things kind of bumping around. So you don't want that. And the larger the pore, the more you start to see these things, the more sensitive the cells. You also see that the cells, these are not native channels to the cells, and they're kind of, the bigger the pore, the more ions and the bigger ions that it lets in, the cell is less happy. So you can have more phototoxicity. And also the cells can like have sort of a window of happiness that is shorter. So carmine, we're also using carmine. That does work pretty well. But some of the more like mutated variants that are much bigger pores, I mean, for me, in our hands, we usually just see that a lot of the cells are dead or just super unhappy. And so finding that sweet spot in your biological prep is really key. And again, we're in ferret visual cortex, which is not necessarily exactly the same as mouse cortex. And of course, if you compare any other brain region between each other, like striatum to cortex or hippocampus or whatever, every area is going to behave a little bit differently. Those cells are going to have a little bit different set of receptors and gene expression profiles. So like you kind of have to find the tool that optimizes across all the different domains for your prep. And that's what I tell people most of the time is like, you find what works for you, and that will allow you to do experiments and then go from there.
Oh, that was really insightful. Good advice. Yeah, it's not as easy as reading the methods part of a paper sometimes makes it seem.
Yeah, there's a lot of tweaking, especially like with the like the photostimulation protocol itself, right? You want to limit average power, but you want to maximize like you want to minimize the time it takes to stimulate the cell. You want to maximize the effect of the laser, but you also, and while minimizing the average power to try to limit phototoxicity as well. So we did a lot of parameter sweeps, and for every new opsin that we use, we kind of have to go through this because they all behave a little bit differently, are expressed a little bit differently, and find that sort of sweet spot.
Great. Yeah, I have a question here. How do you validate the model with the filters to isolate the stimulus evoked response?
Yeah, so standard validation, we're just capturing variance of the responses of the neurons, and then say try to optimize the variance explained. So there's actually a two-step procedure to the fitting. We do a hyperparameter sweep where we fit some parameters and keep them fixed. So like looking for the coupling to the neural pill and looking for photo artifacts and things like that. And then we narrow, and that also narrows in a set of parameters for the visual stimulus and optogenetic filters. And then we do a fine-tuning optimization that tries to fit those filters as best as possible to the response to try to maximize variance. And on average, we're capturing, across our population, we get about 40 to 80% of the variance explained. So, 80 is very good. Usually, I think the sort of back of the hand number that people are okay with for general GLMs of this condition is like 30 to 40%. So we're pretty happy with the 40 to 80.
Yeah, that's great. Okay, we are basically at the end here. So that's really it for questions. So thank you again, Ben, for joining us. That was, like I said, that was fantastic. I was really excited to see what you're up to.